metabolic models of a variety of organisms important in bioenergy research. Other work in the group involves 13C metabolic flux analysis of both heterotrophic and photoautotrophic microorganisms, as well as the stress adaptation of microorganisms to increase growth and product formation rates under industrially relevant conditions. He has published more than 40 research articles in variety of different peer-reviewed international journals, and he also has 2 patents. He is a review editor in systems microbiology in Frontiers group of journals, and he is also an associate editor in Springer Nature Applied Sciences. He is a member of the Technical Expert Committee on Energy, Environment and Forest Biotechnology, Department of Biotechnology, as well as several institutional committees as well. His group is regularly involved in the ICGB training programs pertaining to bioenergy and systems biology. And prior to joining ICGB, he conducted his postdoctoral research work in the National Institutes of Health, NIH, in metabolism in the lab of Dr. Richard Veach. Dr. Veach had worked extensively with Hans Krab, who had discovered the Krebs cycle. Dr. Srivastava published 5 papers during his postdoc and won Fellows Award for Research Excellence FAIR for his research. He earned his PhD in chemical engineering from Michigan State University, and at MSU he researched differential toxicity of saturated and unsaturated fatty acids in human hepatomer cell lines using systems biology approach. He published 7 research articles and won the Sigma Z award for excellence in graduate studies. He had earned his master's in chemical engineering from IISc Bangalore, where he published 3 research articles in international peer-reviewed journals in the field of non-aqueous enzymatic synthesis. So with this introduction, I will now invite Dr. Shirish to deliver his talk. Dr. Shirish.
Good afternoon, everyone. First of all, thank you, Professor Sani and Professor Singh, for inviting me to present the work that we are doing at ICGB in this area. And my apologies to all of you for the delay in my arrival. I started at around 7:15 from Delhi, but Delhi traffic and the road conditions did not allow me to reach on time. So again, sorry for that. So today I'll talk about some insights from life cycle analysis studies of biofuels and what we have done to improve the biofuel research through process development and systems biology. And again, I think it's a very good opportunity for you guys because this life cycle analysis is a very integral part these days of all the sustainability studies. So anytime we talk about sustainability, life cycle analysis becomes an integral part of it, without which obviously any claims of sustainability uh, are not complete. So I think for all of you who are interested in bioenergy research and sustainability, this is an excellent exposure that is being given to you. So please appreciate it. Do I go like this? What's the fastest way? All right. So unfortunately, because of the transfer of files and all, the fonts have become a little messed up. Acha, okay, this is good. Uh, all right. So, but anyway, let me talk a little bit about ICGB, the institute from where I have come. How many of you have heard about ICGB? Okay, a lot of you, but not a lot as many as I was expecting, nonetheless. So, ICGB is an independent intergovernmental organization, which means there are about 68 member countries which have come together and kind of made this institute uh, to solve the problems of developing countries using biotechnology. So there are 68 already signed member states and 18 signatory states. Means their membership is pending, but they have conceptually agreed to be part of this intergovernmental organization. We were earlier part of the UN system, but now we have become independent of it. So our goal is to, of course, to promote research excellence, training, technology transfer, and contribute in concrete terms to sustainable global development. We overall we have more than 45 laboratories in three components we call. Uh, the our headquarters are in Trieste, Italy. Then we also have a component, a very small component in Cape Town, and another very major one, of course, in New Delhi. So, and then there are other components are also coming up, for example, in China and all. So the idea here is to do research which pertains to uh, training people from developing countries, providing biotechnological solutions, and so on. So, not only we do a lot of research in our own laboratories, we also do a lot of support to advanced education. Now again, for a lot of you this may be useful information. Like we support for the students from member countries if they want to go and have training in another member country. Now that is the important part, that if you want to go to, let's say, Italy for some particular training and all, we have a lot of short term and long term fellowships for PhD students and postdocs. So and we also organize a lot of meetings, courses, and workshops. For example, like this, there is also a lot of meetings and workshops on various topics at ICGP New Delhi. You can know more about this if you simply Google ICGB meetings and courses, something like that. But this is the actual link where you will find what are the upcoming workshops we are doing. Now, for slightly advanced researchers, we also have a lot of competitive research grants, uh, and which also includes an early career return grant. So, for example, you were doing your postdoc in some outside India, now you're coming back to India and setting up your own lab. You can have these small but fair amount of research grants to help you come back and establish your lab. And then, of course, we do a lot of work in terms of technology transfer to industry as well as bioethics and biosafety. So, before I begin, I'll talk briefly about this Bio E3 policy. How many of you are aware of it? Though no one is aware of this Bio E3 policy. Okay, now this is supposed to be a game changer in terms of making India a kind of a global biotechnology manufacturing hub. So the vision of the government is, just like in 2000s there was this IT revolution which helped give a lot of opportunities and made India very technologically advanced. For example, a lot of systems nowadays that you are seeing, UPI, etc., they are eventually as a result of our work since 2000 towards developing our IT industry. Similarly, Indian government had in, I think, in September 2024, they have announced this Bio E3 stands for economy, environment, and employment. So the idea is that the next driver for the economy for India is going to be, of course, environment and employment through biotechnology. Now, again, the good part is that we are looking very carefully towards providing a lead in sustainable biotechnology, addressing climate change, food security. Now, uh, the idea here is, and again, sorry for the messed up fonts, but because of the file transfer and all that has happened. So the idea here is to, there are a lot of different biotechnology and biomanufacturing hubs that will be created in India. whose goal is they, for example, there can be what we call biofoundries. Biofoundries is where you will make different components, and then you'll have biomanufacturing. So for example, you have some technology and you want to take it to slightly higher level, you will be contacting these biomanufacturing hubs. So now the vision is, for example, if you have want to engineer something, then you don't really have to go, doing everything yourself. There are these biofoundries which can help you, for example, design your engineering strategies and help you with construct designing and everything, so that you get your proof of concept, and then from there you go to biomanufacturing hubs for scale up and making this product at large scale. So the idea is, of course, encouragement of a lot of bio entrepreneurs, which obviously will be you guys. Uh, so uh, again, when we talk about sustainability, there are different aspects of sustainability. Of course, one is local sustainability of the process that we are talking about. So, for example, when we talk about sugarcane derived ethanol, uh, its its acceptability and sustainability in Brazil versus in India. We already know that Brazil has more than 20%, 25% of its petrol which is blended with ethanol. All of it, almost all of it, is coming from what we can call sugarcane uh, juice. So there, because you have so much land, so much uh, sugarcane production, compared to India, where there is a lot of requirement for sugarcane as making sugar per se. So the acceptability varies, sustainability varies from place to place. Now again, that is a very important component when we are doing life cycle analysis as well. In terms of whatever is sustainable in one location will not be sustainable, exactly same thing will not be sustainable in another location. Then of course we have economical sustainability, which until now that's what most of the industrialists would use to think about, that okay, if I put, I do not know, 100 crores in this project, what is going to be my ROI, return on investment, in how many days or how many years am I going to get my money back, and so on. So they were least interested or less interested in making the process environmentally sustainable. Their goal was economic sustainability. Can this keep on going? Can I capture the market? Can I make money? Of course, these days uh, there is a lot of emphasis on environmental sustainability and also social sustainability. What do I mean by that? I mean that whether the, and in this case we are talking about biotechnological processes, whether the biotechnological process is environmentally sustainable. A very unsustainable process that we talk about is of course agricultural residue burning, which happens in this part of the world or this part of India, and a lot of other places. But obviously, we understand that this is an unsustainable practice. It needs to go, but and the challenge is to find solutions to make this from unsustainable to sustainable. And of course, there is social sustainability also. So, for example, when we talk about different biotechnological, if you're trying to set up something somewhere. So for example, uh, just recently I attended a meeting where they were thinking about growing edible seaweeds in India on a large scale, and one of the challenges they are facing is from fishermen, because they say that if you grow on a very large scale these edible seaweeds, they may have international value, everything else is very good about it, but we were traditionally doing fishing. So that is our main and established way of life, and that's going to be affected. So kind of interaction with local people, uh, so whatever you make has to eventually benefit the local people as well to get you the social sustainability. Now, again, a lot of you may know already about this, that obviously biofuels are characterized in different generations. You have 1G, which is basically what Brazil is doing, making, and a lot of most of the ethanol in India also is produced through 1G, which is basically your sugar-rich substrates, um, which is molasses, um, cornstarch, and so starchy and sugar-rich substrates are fermented to make ethanol, and which is then distilled to make your first generation ethanol. Now, we understand that for Indian requirement, which is obviously increasing, and our targets of also of around 20% ethanol blending, we may not have enough sugar-rich substrates to convert to supply us what we need in terms of 1G ethanol to to make, let's say, 20% blending in petrol. And of course there is this other problem which we talked about in terms of burning of agricultural residues. A lot of residue is already there, can we utilize that which gives you your 2G ethanol. So and then of course the coming up technologies, 3G versus 4G, as well as 3G is primarily growing algae and cyanobacteria in these large um raceway ponds, and using that as the biomass to make biodiesel, and also using converting it to make ethanol and so on. And lastly 4G is where you will engineer these organisms to improve their properties, either growth rates or sugar content, ease of hydrolysis and so on. So that will become your fourth generation ethanol. But in today's talk, when I talk about life cycle analysis, I'll primarily be talking only about 1G and 2G, because 1G is currently being employed at a lot of places, and 2G is what is coming up in India. Also there are a lot of 2G demonstration plants that are being set up. So those are the two currently relevant, but you must know that future people are also very carefully looking into 3G and 4G ethanol as well, or 4G biofuels as well. Now, in order for any biotechnological process to be sustainable, it has to meet these three requirements. It has to be what I call right, RYT, not RIGHT. The right means it should have a good rate, means you can make this product at a good rate, otherwise it will not be competitive. Eventually it comes down to how much of this volume we can make in, let's say, 1 liter, or 1 year or 1 month. So you would always see that even in techno-economic analysis, you would always see plants listed. When I say plants, I mean manufacturing plants, or factories listed as, let's say, million liters per year, something like that. Now that million liters per year is how much we can make in one year, right? So I can put a plant which is 50 million liters of ethanol in a year. Now, how much I can make is a function of two things: the size of the equipment, and also how much I can make, how fast I can make. Because if I can, let's say fermentation, typical fermentation takes around 3 days with one process, and in the other process it takes just 2 days. So your productivity, which means the amount that you can make in 1 year, is suddenly increased one third, which is a huge amount. So of course you want processes which are fast. Very important thing is yield. Yield is defined as how much product you can make per gram of substrate provided. So in case of say, let's say 1G ethanol, primary substrate is sucrose and primary product is ethanol. So if I give you 1 gram of sucrose, what is the amount of ethanol I am going to get back? Because that tells me how efficient the process is. And again, we obviously want to aim for something on the order of 90% efficiency and all over, just because the less efficient your process is, which gives you two things: mainly either the organism is not completely utilizing the sugar, or the there are other products which are unnecessary for our particular case, which the organism is making. And either of these scenarios is obviously not good. So yield is very important. And now there is also, I will not talk about this, but there, for all the sugars and lot of products, there is something called theoretical maximum yield. So for example, for ethanol production from glucose, let's say, the maximum you can get is 0.51. You cannot get more than 0.51 gram of ethanol per gram of glucose. All right. So you always, most of the industrial processes try to be close to that number. Most of them you will see is more than 0.45 gram ethanol per gram of sugar, something on that order. And the final thing is the product titers, which means again how much of this product I have per liter of my broth, because that eventually controls how much. So effectively, all biotechnological processes, especially those which are done by microbes, have a lot of water in it. And then the ethanol that you want is, you want anhydrous ethanol. So to remove water from an anhydrous ethanol, you want to obviously want to have as much ethanol in your broth as possible. So that becomes your titer. Uh, now for a lot of again, you want something of the order of 20 to 25, 20% or so of your product ethanol in your broth. And this all obviously requires a lot of engineering, lot of efforts in identifying robust organisms. So we'll talk about LCA of 2G, LCA of ethanols. Now, when we talk about 2G ethanol, okay, yeah, when we talk about 2G ethanol, we start with a lignocellulosic feedstock. By the way, this figure is taken from International Energy Agency. So this is a simplified diagram where we take it through pretreatment, then through enzymatic hydrolysis. Now, enzyme can be produced on site generally. That's what is recommended, but also can also be imported by or supplied by certain manufacturers. And then from the sugars that are released, you go for fermentation, then you do distillation, separate the solids, and all this waste are, then you can kind of burn that waste to generate electricity, which we call cogeneration. And of course you have separation of ethanol, which is then further dehydrated to make your anhydrous ethanol. So that's your simplified diagram. A lot of other processes occur in between. Now, this review which came in 2009, now I understand it's an old review, but it tells you what are the challenges of biofuel crops, for example. And it also talks about different um, biofuel crops and what are their, let's say, net energy value. Now, net energy value is how much energy I'm going to spend to grow that crop versus how much energy I'm going to get if I burn the crop or the product. So according to, and again, understand that this is also relevant to particular type of corn, particular. So according to this, corn with stover will give you the highest net energy. Now, of course, this study is based out of US, where there is a lot of corn grown, and so they say that corn along with stover can give you the very high net energy. But again, the in terms of what are the major insights from all this particular study, what they kind of um, concluded was that there are hundreds of studies of LC, and again, it's possible that other speakers before me have also talked about this, but there is a lot of variability depending on what are the various parameters you choose. So of course, the factors that implement uh, or impact the LCA results are, what are the system boundaries that we take, okay, what are the different life cycle inventories that we take, and of course what are the efficiency terms and functional units that we do. So based on all these variations, you can have one study which says that the, let's say, biofuels are significantly carbon negative, and the other will come out to say no, they are may even be slightly carbon positive, which means you are generating carbon when you are burning um, biofuel. So there is a lot of, again, it's almost like the experiments you do in your lab. One person will come and say, oh, I always get this titers and this ethanol and all that, and the other student will come and they will do it probably slightly differently, and then they will get a completely different titer or profile, growth profile. So again, it depends on your assumptions and the values that you take. Your results can be varied. But the good part of LCAs is that generally, almost always, you get a range of values which tells you kind of your confidence level as to what parameters are less divergent and which are more divergent. I'm sorry again. So this is this another study which looked into comparison of different feedstock for ethanol production. And this was, again, this study was in Mexico. So they looked at various lignocellulosic biomass, and they looked at mainly their environmental rank of the fuel that is produced. So if we use this feedstock for making ethanol, they what is my, let's say, CO2 savings, and based on that they gave it an environmental rank, and also what is the cost of my production of this going to be. So in this work, obviously the very important thing to notice here is that and they looked primarily, they found that mainly, obviously, agricultural residues and woody residues are the ones which are most environmentally important, or which have the highest environmental score, which means that if you use agricultural residues and wood waste, then you can have the maximum environmental impact. However, the more important thing here is that the environmental rank does not correlate with the economic rank. Economic rank deals with what is the cost of production of ethanol, let's say. So obviously, if you, again, in Mexico, if you use rice husk, you can have a lot of environmental benefits, but um, the cost in terms of relative production will be highest, will be very high. On the contrary, if you use, let's say, wheat straw, your cost of ethanol production would be very low, though your environmental rank does not is very poor. So the question here, obviously, there is this tricky question that comes out, is that it's not a one-to-one correlation that if I can make the cheapest ethanol, then it would be most environmental friendly, or vice versa. So we need to find out some sort of balance which gives us both environmental benefit as well as economic stability. And again, ob- now the second conclusion is somewhat obvious, is that the biomass composition impact the result. So they looked at mainly ethanol production as well as cogeneration of electricity, and they found that mainly, of course, the ethanol production is related to the cellulose content or the sugar content of the biomass, and while the electricity that is generated is related to the lignin content. So, of course, there is this again balancing of how much sugar and how much lignin is there in your feedstock, and they also found that availability of feedstock is very important. So, for example, rice husk may be a very good environmentally friendly crop, but then its availability is not throughout the year. So generally, when you want to design biofuel kind of processes, they have to be somewhat agnostic to uh, the feedstock that is used. So another study looked at now ethanol from poplar. Poplar is your tree, right? So the woody biomass, the advantage there is it grows fast, so you can also harvest it in 2 to 3 years, or 5 years is the maximum generally, and you get this woody biomass which can be then converted to ethanol using somewhat similar process as what I have shown. And they looked at, and what they found out was obviously growing those poplar trees had the biggest environmental aspect impact. About 40% of the environmental impacts were just based on the utilization of all these chemicals and water for growth of these poplar. And again, so they also tested the impact of GM, genetically modified poplar, which has lower lignin content and higher sugar content. So, and again, they found that this would be really beneficial. So on a long term basis, eventually it may be beneficial to use these GM poplar uh, because the amount of ethanol that you get is much higher. And also they found that at the biorefinery stage, it's the use of cellulase enzyme that dominated the environmental burden. So, and again, if you look at the various studies with 2G ethanol, you will find this cellulase enzyme being limiting both for in terms of economics, as well as you see here, they also have a very high environmental burden. So, and then they found that obviously achieving higher ethanol yields per unit of enzyme. Now again, enzyme becomes important without introducing chemically intensive pretreatments will be essential to reduce the environmental profile. Now generally what you do is you treat your biomass, you first of all you grind it up to a particular particle size, then you treat it with acid or dilute acid or ammonia or other harsh chemicals. There are other very lot of different research going on and different treatments. Anyway, so they found obviously that you need to have um, less chemically intensive treatments, and generally something like steam explosion is what is being recommended these days. Then there is this another study where they compared 1G and 2G ethanol from corn grain and corn stover, and this again important is that this was done for California, USA, because uh, if you understand, most of the corn that is produced in US is in their central states, and this California is, I don't know, more than like 1,000, 2,000 kilometers away from there. So the idea is that some of the uh, corn that is grown in California will be combined, and then these corn and corn stover will come in train to this California plant, and they will make ethanol from that. So I'll not go into all the details what they found, that but their major insights was that displacement of cattle feed by this wet distillers grain and solubles. Now after the fermentation happens, especially with grain ethanol, so you take this grain and then you kind of get the sugars and you ferment, so whatever is remaining is actually a very good feed for animals. And in California there is a big farm kind of system where they grow a lot of cattle and all, both for milk purpose as well as meat purpose. So they say that obviously if you set up such a plant which makes 2G ethanol, then you can also have and for these cattle rearing, they get their feed imported from these Midwest region, and this DDGS that comes from again 2,000 kilometers away, so that the price is very slightly higher. Now if you can make that ethanol locally, then obviously a change in the uh, price of this WDGS uh, will have a significant impact on GHG emission. The However, the assumption here is that the WDGS means uh, the residue that is left after your fermentation, which is, so you can ferment grain or you can ferment corn stover. Okay, corn stover is the, you can say, the waste part of the corn, right? So the assumption here is that the uh, WDGS that you get from grain fermentation is nutritionally and cost wise the same as the WDGS that you get from corn stover fermentation. Now again, that is a assumption that can be questionable, but anyway. And again, if you look here also, you see that cellulases are the second most important factor in terms of their 1% change in their cost can have more than 2% change in the GHG emissions and so on. So, sorry. So again, understanding that okay, cellulases play very important role. At ICGB, we have been working on developing our own cellulases. So we have been working with process development for improving cellulase production. Now, obviously, the idea is that after pre-treatment, you get enzymatic hydrolysis, which releases sugars and which can then actually not only make biofuels but can also make other products, for example, biochemicals, biodegradable plastics, so on and so forth. So, and if you look at cellulase cost per se, it costs around 15% of your ethanol cost. So, if you make one, say, 100 rupee cost of ethanol per liter, then 15 rupees effectively coming from the cost of cellulase that is used in that production. So at ICGB, Dr. Shamsi Astani's group has identified a fungus with high cellulase production and also engineered it to increase its enzyme production and reduce inhibition in presence of glucose. So generally what happens is cellulase production is very sensitive to availability of glucose, and biomass likes glucose. So if you want to grow biomass, you kind of need to provide glucose, but once the biomass is growing, cellulase production goes down just because of the strong inhibition. And at shake flask level, we did a lot of optimization, so which led to titers of about 3.5 FU per ml with about 11 to 14 gram per liter of protein. Now again, all these values are very important when you are, so this is your, in a sense, titers that we are talking about. And then what we did was we designed a fed batch feeding to improve the production to from 3.5 to 5.5, and protein also was increased to up to 18 to 20 grams per liter at 20-liter scale. So again, all these terms are important. What is the product title, what scale we are talking about. So sorry. So with the success of that, we went for scale up of this cellulase production, which means we went for making more of it. So this was done again at a biotech company with fermenters of required size. So we scaled it up to 15,000 liters. Of course, ICGB does not have those fermenters. So we went and talked to this company and we scaled it up to 15,000 liters. It has its own challenges. So again, when we talk about science, many times we don't talk about these challenges. So it required a regulatory approval because this, we are talking about a genetically modified organism. Uh, Beyond a particular, so anyway, when we are talking about a genetically modified organism, even at a lab scale, there are certain regulatory requirements, but especially if you are scaling up, there are very stringent requirements that needs to be followed up. um, and also, since this was to be done at a secondary place, that we needed to really prepare a sufficiently detailed SOP. So which means the protocol has to be very carefully written, every little detail of how the spores will be prepared, how the pre-culture would be done, at what hour you would harvest, and so on. So very detailed SOP needed to be. And then we talk about logistics. So of course, if you're talking about 15,000 liter fermenter, you're talking about hundreds of kilo of substrates and things like that, which has to be delivered to the site where it is to be produced, and then coordinate with these guys so that when we order it from that company, it is something is reaching there which is 100 150 kilograms, so they must be there to receive it, and so on. So those are some of the practical challenges. Now at that scale, uh, there are other things which becomes important, such as preparation of simple solutions as well as sterilization. So in lab, many times what we do is, okay, I have to prepare a solution. I pour water in a beaker and put my thing, put it on stirrer, and it's okay. Everything is okay. But if you're talking about at that scale, obviously preparing solution takes a lot of time. Sterilization and then cooling can take, at 15,000 liter scale, it can take up to 10 to 12 hours. Okay, so all that thing has to be carefully thought about. We even have to tell them how many times we need the fermenter to be washed, because each washing cycle is about 10,000 liters of water. So all those little things become very important. So anyway, we did an initial test at 200 liter scale, and the good part was we could reproduce what we were getting at the shake flask level at the lab, which gave us the confidence. So we didn't directly go from, of course, 20 liter to 15,000 liter. You always go to a middle stage at a new place, and then later on we got it at 15,000 liter scale. Again, over 5 FPU was got, and about 16 gram per liter protein was got. Now, in terms of, so it was a fed batch fermentation. Anytime you want to make something at higher concentration, we generally go for fed batch fermentation. So we had kind of, now this is pink is showing the biomass. So we aimed to have a fair amount of biomass as soon as possible. So this initial feeding phase was designed so as to have uh, high biomass, and then we kind of maintained that biomass and induced it with different inducers to get the production of the cellulase. Now again, the good part was our production profile at 20,000 liter scale looked exactly similar at what we were getting at 20 liter scale. So that this shows that this process is quite scalable as long as you maintain the quality. So then obviously another important thing when we talk about industrial scale is downstream processing, which basically where we could get it to fair amount of FPU at high protein concentration, and then this also was a challenge at that scale, because the company that we worked with was not really looking at fungal fermentation. They were looking at yeast fermentation, and these kind of small things can make a big difference at industrial scale. Anyway, so this was a kind of a success story for us, where we could take something from the lab and scale it to higher levels and kind of maintain the similar production profile. So what I'll talk also about is that obviously the biggest challenge with biofuels is that it's a low value commodity with high capex and opex. Means it will require a lot of money to set it up and get the process running on a regular basis, and that's why you hear about the values of something on the order of, if you I make ethanol, currently my best technology is roughly on the order of 130 to 150 rupees a liter in India, which of course you're not going to pay for, right? So the point is that one solution is we can go for biorefineries, which can give you more than one product. Traditionally, in terms of biorefinery, people have not done anything. The lignin or the byproduct that they get, they simply burn it and get energy from it. So that's what their co-product is. But obviously, what we are talking about is that you can have a lot of other products as well, and which can further increase the sustainability. And the same thing is done whether are looking for lignocellulosic biomass or algal cyanobacterial biomass. At the end of the day, things are made up of sugar, lipids, and proteins, which can be then further utilized for other products. Uh, However, if you want to biotechnologically make products, it has to go through something called a DBTL cycle: design, build, test, and learn. So we do this metabolic engineering, we try to make a product, and then we test how much product is formed, or if the organism is not performing, we learn, and then we go through this cycle many times, so as to get a final organism which can do what we are talking about. And again, it's very important for us to understand that in theory, at least, the metabolic pathways have solutions to a lot of our problems. We talk about plastic pollution, there is a solution. We talk about CO2 pollution, there is a solution in this metabolic pathway repository. And so, for example, we know that we can make products which are direct substitutes for petroleum products or even plastics, and other metabolites can act as precursor for synthesis of other products. However, in order to make these products at a reasonable scale, we need to do metabolic engineerings many times. So again, the metabolic chassis is something that can help you identify um, the engineering that is needed to overproduce the compound that you are looking for. Now, and in that sense, I'll introduce you to metabolic networks, which are very similar to roadmaps for cellular processes. So, for example, if I'm coming from Delhi, I put a roadmap which told me what is the fastest way of uh, reaching this place. Of course, there were challenges in it. So Google Map is also improving eventually, and many times it's introducing these things. So Google Map is also going through DBTL cycle, right? It's updating the road information where there is road construction, trying to tell you, and then real time traffic information is being inbuilt and so on. Exactly similarly, for us, we are also trying to make not only roadmap but also real time information of protein concentrations, the if we have any other flux measurements, things like those, in order to improve the accuracy of these metabolic models. So our lab works a lot of on genome scale metabolic models, as the name implies. Um, Basically, what we do is it's a model, which means it can help you simulate cellular responses with metabolic reactions. A lot of almost all metabolic reactions within a cell. Now, almost all is a little bit of overclaim, but still we want to have in our genome scale metabolic model most of the reactions of, or at least the major, almost all the major pathways of the organism. And this has complete pathways. The model must have complete pathways so that the precursors can be synthesized. It also has something called gene protein reaction relationship, which tells you, for example, which gene or set of genes code for which protein or complex of proteins which constitute which reaction. And it also contains something called a biomass equation, which tells you what are the individual components that are needed. So how much protein is needed if you want to make 1 gram of biomass, and within the protein how much is the composition of different amino acids. Similarly, I do it for DNA, I do it for lipids, everything. So this is a even though I have written just one sentence, there enough information goes on in there. It also has mass balance reactions with defined directionalities and defined uh, location of reactions. For if you are talking about eukaryotes, we also kind of restrict the reactions into individual compartments. So this is a at the very minimum, this is a very good repository of metabolic information about the organism. You know exactly what all genes are involved in what all processes. It starts with a genome sequence. Of course, the genome sequence we need to assemble and annotate, and then go for what we call draft reconstruction. Draft reconstruction is okay. from the genome annotation, I know that okay, I can identify these genes, so some of these pathways I understand should be there. In any case, sometimes, many times, we also know that this pathway has to be there. So for example, if we are talking about phototrophs, the pathways for CBB cycle, or pathways for, let's say, the simplest scenario, synthesis of these chlorophyll A and B must be there, right? And synthesis of chlorophyll A and B itself is a long pathway. So again, um, we go from draft reconstruction to a big cycle of refinement of the model of the draft. At each point, we test our model for growth, biomass formation, growth on known substrate, known lethal knockouts, and so on and so forth. So we try to make our model as close to what really happens as possible. Once you make that, of course, you can identify what are the unique enzymes and isozymes and pathways that are in my organism. We can predict the growth and flux distribution under different conditions: autotrophic, mixotrophic, heterotrophic, and other substrate, what it can make. We can identify bottlenecks in the use of particular substrate. So for example, if one organism is unable to use, let's say, any substrate, what reactions can we add so as to make it use that substrate. I'll talk about it a little bit more. Uh, we can identify reactions which are important for growth, identify the effect of knockout over expression of heterologous pathways. So again, when you are designing these synthetic biology modules, ideally you want to know that okay, what is the potential impact of these introduction on my organism. And of course, genetic engineering strategies to improve metabolite production. I'll talk about it briefly. So the second example to talk about is utilization of these metabolic model to, in this case, improve alkane and fatty alcohol production in E. coli. So in E. coli, if you want to make fatty alcohol and fatty alkane, you need to introduce these ADO, AHR, ADO or AHR enzymes from heterologous sources. Okay, so for example, in this particular studies, um, this were enzyme for alkane synthesis were taken from cyanobacteria and expressed in E. coli. All right, and then it was also found that this um, basically AAR, what it does is, so AAR and ADO were introduced. AAR converts fatty acid ACP to fatty aldehydes. Now the aim is to make more of this, but we also found that it also has these native AHRs, aldehyde reductases, that leads to formation of fatty alcohols. So now eventually what we want to know make is a combination of these two. And we also found with various studies that moderate expression of AHR improves alkane production, again through redox balance and so on. So we used metabolic modeling to understand what are the various reactions that lead to formation of alkane. In this case, it's written here. Again, the resolution is not good here, unfortunately. Um, and we added these heterologous reactions for alkane synthesis from cyanobacteria. So in this model, we added these reactions which are shown in pink here, which are the reactions which taken from cyanobacteria and introduced in E. coli. And we identified the reactions associated with increased alkane production by a mathematical modeling technique called flux scanning through enforced objective function. So in which we effectively um, in, increase the production of our desired molecule while trying to minimize the effect of on growth, and then we see what are the metabolic reactions whose flux has to change. So it's almost like a sensitivity analysis, if you guys are aware of. So our modeling showed that effectively what we needed to do was we needed to increase the expression of this ZWF. ZWF is the gene which, or the protein which converts this glucose 6 phosphate to this first metabolite in the pentose phosphate cycle, and this has to be increased, and then we needed to remove 7 byproduct formation reaction to increase the yield. And but not only that, this was also tested in various E. coli strains to see which gives you more yield. And interestingly, the DH5 alpha showed initially the highest alkane production, so this was chosen for further engineering. And by putting the model predictions, we went from 115 milligram per liter to 318 milligram per liter alkane and 1 gram per liter of fatty alcohol. And by using fed batch fermentation again, here we produced more than 2 grams of 2.5 grams of alkane and more than 12 grams of long chain fatty alcohol. So these were the maximum titers of hydrocarbons reported in any microbe. So this showed the power of metabolic modeling to improve the product titers. The other example I'll talk about briefly is production or metabolism of xylose by Saccharomyces cerevisiae. Now you may know that S. cerevisiae does not natively ferment xylose, which is your major challenge when you are talking about 2G ethanol. How do I engineer? And there are these two pathways to ferment, to metabolize xylose in sacro. Excuse me. One is this XI xylose isomerase, and the other one is XRXDH pathway. So and then so basically, obviously you know that about 20 to 30% of sugars in lignocellulosic hydrolysate is C5 or xylose, combination of xylose and arabinose. So our idea was, what are the various engineering that we can do to improve the xylose fermentation in sacro. So we used now again here it is very interesting to know that for example, for a lot of industrially important organisms, say E. coli, as well as more importantly for yeast sacro, let's say, there are these, it's not that once you make one metabolic genome scale metabolic model that it ends there. For sacro, these are all individual yeast metabolic models that have been done. So it started with one model which came out in 2003, and then people worked on it and improving it and so on and so forth. Multiple rounds and multiple cycles of improvement were done, where now we have these metabolic models which we call enzyme constrained metabolic models, which means there is this proteomic information that is built in to further uh, improve the predicting capability of the metabolic models. And so we use this yeast 8 model, which again which consists of almost 3900 metabolic reactions, 2700 metabolites, and about 900 genes. So and again this was at that time was the most advanced model. So we use that, and we compared the flux distribution by flux balance analysis, which is a computational technique, to what are the published values of metabolic flux analysis. So this tells me, FBA tells me what is the flux that is needed through various pathways in order to utilize this, and MFA tells me what is the actual flux that is happening in the organism. So ideally, if you want to utilize it best, your theoretical and actual fluxes should be very close. So and again here in this case xylose intake was constrained to published experimental values, and we use this Cobra toolbox. So what we found was that the flux, FBA predicted flux and 13C predicted flux, were fairly close for upper and lower glycolysis, which means your upper and lower glycolysis do not need any much modification. But if you look at PPP pathway, the required flux is about 34% of the carbon that is going in, and the actual measured flux is about 19%. So obviously this becomes your immediate target which tells you that you need to improve the activity of PPP in order to be able to utilize your xylose better. So, and within this, we also found that RPI, ribose isomerase, and transcriptolase were the targets. And though we did not experimentally validate, this was very well corroborated with subsequent literature results, which says that basically this 413 is a regulator whose deletion increases the activity of pentose phosphate pathway, and of course it improves the xylose metabolism. Um, so this corroborated with what we found. We also tested the response of the model to in presence of various inhibitors, because that's again something that impacts ethanol production. And what again we found here was basically that again NADPH production is the limiting response to furfural inhibition. So here also, if you want to um, get your cells which are robust and against inhibitors, you need the NADPH cycle or pentose phosphate pathway activity to be higher. And natively, sacro is such a way that almost exclusively glycolysis happens in that. So, in order to utilize this strain for xylose fermentation and inhibitor resistance, you need to have higher PPP activity. So, that's your target which was suggested. The last I will talk about is citric acid production from bagasse hydrolysate. So again, we talked about making other products other than, let's say, ethanol. So one possible product is citric acid, because citric acid is almost exclusively produced through fermentation. Generally, A. niger is used, and it has it is used in food, pharmaceutical, and personal care. So a lot of places where there is regulation of pH, it requires citric acid, and its global market is quite high. Now, no surprise there is that China is the biggest supplier for citric acid to the world, and A. niger is your primary fungus that is used for fermentation. Uh, however, the problem with A. niger, in subsequent studies, it was found that it produces certain fungal toxins in its biomass. So ideally, for any biotechnological process, you need to be able to utilize the remaining biomass. Okay, for example, we talked about WDGS, right, which is the remaining from biomass of yeast, as well as the remaining biomass from the fermentation, which adds value to your product. But main product is ethanol, but I sell this yeast biomass also, which is used as feed in order to make the process more economically viable. But A. niger uh, produces certain fungal toxins, so you cannot really use it as a feed for other animals. You literally have to burn it. So this was we worked on this international collaborative project, where our collaborator has identified a non-toxinogenic citric acid product producer, so which is called A. tubengensis. And this however, obviously this is not your um, standard used organism, so very little metabolic information was available. So what we did was, we reconstructed from its genome sequence, we reconstructed its genome scale metabolic model in order to understand what are the pathways that are active in this under different conditions. So it started, as I said, with draft model reconstruction, utilizing both A. niger as well as A. tubengensis available genome annotations, and which led us to this draft model. And then also utilizing the information of our own strain, we further refined. And as I have told you guys, we did a lot of refining of this model, including measurement of its biomass composition, its rate of substrate intake, rate of growth, everything was measured. um, we did a lot of this, which is called biolog plate assay. This biolog plate assay, what it does is it kind of grows the organism in about 94 or 95 different substrates. So this is a 96 well plates, and we can grow them on all of these substrates to see how the organism will grow in presence of different substrate. So we incorporated all this information in our metabolic model, so that now our metabolic model kind of reproduces, at least in terms of growth, how what happens with different carbon substrate. And then we used other bioinformatic tools um, to kind of define which reaction occurs in various compartments. So we have this peroxisome, we have mitochondria, we have cytosol, and we have extracellular compartment. So we had in our model these 4 different compartments and have reactions which are specific to each compartment and so on. So again, this model consists of 1652 genes from this organism. These are metabolic genes, 1657 metabolites, and over 2000 reactions, which were distributed in 4 compartments. And as you can see, so these dots were the measured fermentation profile. So this is how the biomass growth looks, and this is how our model captures the biomass in this line. And then this is how the phosphate intake experimental data looks, and this is how the model captures citric acid production, glucose consumption, and xylose consumption. So as you can see, this large scale metabolic model is able to capture uh, the fermentation profile for citric acid production by this new organism. Now obviously this, once now this is made, this offers a framework to conduct further systems biology analysis. So overall conclusion, what I would say is that obviously LCA results, as I mentioned, are impacted by several factors. So whenever we are designing and conducting LCA studies, these small details have to be very carefully justified, is probably the correct word, is that you need to, whatever parameters that you take, you need to have a justification. Um Now, from the LCA studies, it is known that cellulases are very important for 2G ethanol production, and I gave you an example where we scaled up the cellulase production with an indigenous strain to 15,000 liters with good activity. And when we talk about xylose metabolism as well as inhibitor detoxification, PPP activity is very important. Now again, this is important not for both xylose metabolism, furfural detoxification, as well as fatty acid production by E. coli. And it is possible to achieve near theoretical yields of biofuel molecules. However, this will require several rounds of engineering. And non-model strains can be useful in bioenergy research, and genome scale metabolic models provide a useful framework to conduct their metabolic systems biology research. So with that, I would like to thank obviously the funders, Department of Biotechnology, BBSRC, for supporting our research, collaborators, Dr. Yasani, as well as Professor Fell, Mark Pullman, and Jamie Wood. So thank you all for your attention.
Thank you, Dr. Shirish. So we now have this session open for discussion. We are running short of time, so we can have 1 or 2 short questions.
Yes.
Hello. Thank you for a very elaborative lecture related to cellulose and metabolic engineering models. I have a very short question related to cellulose.
Yeah.
What is the stability of the cellulose under industrial operating conditions after
Right.
you produce, and what are the strategies one can look for to improve the stability and activity?
Right. So we tested this cellulase, and at least at 4 degrees C storage, these are stable for couple of, 2-3 months. So at least until that time we have stored. In terms of improving the stability, first of all, obviously, you need to design your feeding strategy so that you are not feeding too much of protein and just balancing the right amount of protein, so that you don't produce too much of proteases. Because what happens is, Industrially, these enzymes will be concentrated and kept at generally at room temperature. And so there, if there is a contaminating protease present, that will degrade your enzyme. And in theory, you can also add some protease inhibitors in order to, these are chemicals which you can add, but generally if you look at protease inhibitors, they are prohibitively costly. So almost certainly there would not be any addition of protease inhibitors, most likely, and that's why you need to kind of design your production so that your protease contamination is as little as possible. You can also change the pH of storage. So for example, if there is acidic proteases that are being produced more, you can make the pH at least for storage basic, so that those are, the activity of those are low. So those are some examples. Thank you.
Any questions? Anybody having any more question? If you don't have any questions, so we can close this session. So I thank Dr. Shirish to coming here and delivering this lecture, where he talked about this systems biology and metabolic pathways, how you can design your experiments and how you can use it to enhance the yield of like different bioproducts in biorefinery. Okay. Thank you, Doctor.
Thank you. Thanks.